Welcome to the final lecture in the Basics of Neuroscience course. First, I want to say congratulations for completing the course. I hope that it exceeded your expectations and that I was able to give you a solid foundation in neuroscience, and I hope that you were as intrigued and amazed by the brain as I am. In this last lecture, I'm going to cover some of the common research methods and techniques that you may encounter when you start reading research articles. However, it is important to note that not every neuroscientist has a good grasp on every method out there, because there are really so many of them. Now, because of this, there are so many resources available online and on YouTube about any type of research technique. So if you ever see a technique that you don't know about and that is mentioned in the materials and methods section of a paper, then you can just do a quick search and you'll probably find a great animation that will show you how the process works and what kind of data it produces. The most important thing to emphasize is that the type of research method that is used will depend on the research question and also usually on the specific branch of neuroscience. So for example, cognitive neuroscience or neuropsychology studies often rely on human participants and techniques that measure brain activity and structures, such as magnetic resonance imaging or MRI, or functional MRI, or EEG recordings that measure brain waves through electrodes that are placed on the skull. There are also many cognitive tests that can be performed. Now, on the other hand, in the fields of neurobiology or cellular neuroscience, there is typically a need to measure the expression of certain genes or proteins, and for these, there are various possible methods that I will go over. Now, keep in mind that the techniques are always being improved. So, for example, the gold standard for measuring genes across cells and tissues used to be RNA-seq, which is basically sequencing mRNA transcripts to evaluate the expression of genes. While now, the gold standard for this is single-cell RNA-seq, which evaluates the expression of genes in every individual cell rather than across a cocktail of different cells. So the techniques are always changing, but here I'm giving you the basic overview and the principle, which is usually the same even in newer methods. With classical MRI, it is possible to scan an individual's brain and then analyze different structures and their sizes. In functional MRI or fMRI, the underlying principle is that increased blood flow to a certain area means increased activity in that area, and therefore fMRI will show us the live change in activation of different areas, typically while an individual is performing a certain task. So it is not static like a normal MRI, but rather it allows researchers to look at dynamic changes and activation of areas in response to stimuli. So here I've put an example from a paper in which the research participants were performing a memory task while in an fMRI machine, and we can see the red areas are basically the ones that are activated or have increased blood flow, and they are spread out through different parts of the brain which are known to be involved in memory. For measuring gene expression, a polymerase chain reaction, or PCR, can be performed. There are different types of PCRs depending on the goal, but the principle again is the same. So the more highly expressed a gene is, then the more abundant its mRNA transcripts will be. For a real-time PCR, we can take RNA, use a reverse transcriptase enzyme to convert it into DNA, then perform a PCR reaction. For real-time PCR, we require a fluorescent dye to be added, and it will be detected by a sensor in the machine. The fluorescent dye will bind to DNA, and the signal gets stronger the more copies there are of the gene. In the end, we can use the graphs that are produced by the machine to calculate an approximate expression level of the gene. So this is a diagram showing the different steps of an RT-PCR reaction and also what the possible results could look like. For this, there are a lot of nice animations on YouTube that you can watch to really be able to visualize this process and understand it in detail if you are interested. For measuring proteins or visualizing proteins, we can perform immunostaining, which means we can add certain molecules that will allow us to visualize our protein of interest under a microscope. Immunostaining is based on a specific interaction between an antibody and an antigen. And in this case, the antigen is our protein of interest that we would like to see. So for this, we have to add a primary antibody specific to our protein, then a secondary antibody in order to amplify the signal and make it possible to visualize under a microscope, because the secondary antibody is usually coupled to a marker such as a fluorescent dye for visualization. This is how the process occurs. The primary antibody binds to the protein or the antigen, and then the secondary antibody binds to the primary antibody, and it also amplifies the signal since we have multiple secondary antibodies binding to one primary antibody, and it also allows it to be visualized due to the presence of the fluorophore, which is the fluorescent molecule. Now this is an example of an immunofluorescence staining, and it's actually from my own research publication. So it's possible to label multiple proteins, and this is what we are seeing here. So green is for one protein, and red is for another, and therefore the orange color indicates that there are both proteins present in the same cell. So immunostaining is very valuable because it allows us to visualize proteins in the 3D context of the tissue, and we can therefore also make conclusions about spatial patterns and connections between different proteins. Now, if we don't care about the spatial pattern and we would just like to quantify the expression of a certain protein, then we can perform a Western blot. In this procedure, proteins from a certain tissue are first separated by gel electrophoresis, which is basically applying an electrical current to a gel that results in separation of the proteins according to their size. This happens because we know that larger proteins move slower than small ones, so the result is a gradient of proteins by size. Proteins are then transferred to a membrane that can then be processed similar to an immunostaining experiment. So first we add a primary antibody for our protein, then a secondary antibody, and then we can visualize the resulting bands and make conclusions about the expression of a protein based on the thickness of the bands that we see. So thicker bands means higher expression of the protein. So as a practice exercise for you to start reading articles and getting used to that process, you can check the assignment that I've put up, which requires you to read an article and then answer some questions or basically summarize the article. So thank you for taking this course, and I really hope that you enjoyed it.